Good evening, all. Hello. Hello. Hello.
Yes, sir.
Am I audible to you?
Yes, sir. Audible, sir.
Good
Right.
evening, sir. Good evening,
Very,
sir.
very good evening. Very good evening. Very good evening. Yeah, black for biotechnology 2, transgenic plants, plant genomic, proteomics, plant metabolics, and intellectual property. These are all completed. Now remain only one black, that is black 2, right?
Yes.
Okay.
Sir.
Black 2, molecular biology 2. So first topic, restriction endonuclease. What are the restriction endonucleases? What is the function of endonucleases first? Just wait. Right.
The screen, please, sir. Sir, unable to see the screen. Now clear? Yes, sir.
Restriction endonucleases. What is the main purpose? Restriction enzymes. Restriction enzymes, these are cut the DNA strand and cleave the DNA strand and recognize DNA strand. Majorly, this restriction endonucleases, three functions such as it can recognize the recognition site and can cleave the DNA fragment and can modify DNA sequence. So these three functions, the main three functions of the restriction endonucleases. Restriction endonucleases first discovered by Daniel Nathans, Warner Arbor, and Hamilton Hosmith. They not only discovered the restriction endonuclease, and also these discoveries led to development of recombinant DNA technology. Already told you what is rDNA technology. How this restriction endonucleases involved in rDNA technology. Hello?
Sir, here. Listening, sir.
Yes, what is rDNA? Recombinant DNA. Recombination means
Combining the proteinaceous material which is present in the DNA.
proteinaceous.
Proteins, sir. Proteins. Amino acids and proteins.
No, what is the combination of DNA? I am asking you. I am asking a simple question. How the two DNA combined in
Sir,
general process.
sugar phosphate group, sir.
Okay, sugar phosphate group, that's okay. But whenever these combination formed in organisms,
Nitrogen bases.
and process of timing.
Timing.
Already told you in tissue culture technique,
Yes, sir.
recombination or DNA or chimeric DNA. I have explained clearly in general process when rDNA and how rDNA formed and how artificially recombinant we get and what
Yes,
are the
sir.
benefits of rDNA also. Name itself recombination. Recombination simple. Here this is the one organism, this is another organism. Maybe this is female and this one male. During fertilization, which produces the male gametes, which produces the female gametes. The both are fused during reproduction. What happened here? Zygote formed, right?
Yes, sir.
Now this male, female, both DNA, both the chromosomes are present inside the zygote.
Very good.
Now this is single cell. This single cell undergoes to mitotic division and divide, redivide and form number of cells.
Yes.
Number of daughter identical cells. 2, 4, 16, 32, 64, like uncountable number of mass of group of cells are formed. This group of cells called as embryo. After this embryo also differentiated into plant parts such as root, shoot, and flower, right? Root, shoot, and flower. What happened after formation flower here? The flower is the reproductive organ, right?
Yes, sir.
Can I rub this?
Yes, sir. It undergoes meiosis division again.
Right. That's what I want.
Yes.
So after formation of flower, reproductive organ, which produce the male and female organs such as androecium and gynoecium. In androecium, what happened? Pollen grains are formed. Flower. Flower, androecium, from androecium, pollen grains are formed. As it is from the gynoecium, ovules also formed. But here the formation of these pollen grains or ovules, what happened? Meiosis division takes place, right?
Yes.
As a result of meiotic division, pollen grains are formed. That's why the pollen grains always haploid in state. Ovule also haploid in state. What happened during meiotic division? Before meiotic division, the diploid state, flower, root, shoot, leaf, they are diploid state. In diploid state, the chromosomes appear as a pair. For a blue color from male one and the red color from the female one. It is as a from the group, a giant. These are called bivalent and the state called diploid state. But what happened during meiotic division? Meiotic division, it becomes single, right? That means this one, the blue color separated and red color separated becomes off. That means it became a haploid state. While they are segregated into gametes, they can exchange the material. They can exchange means here the red color portion somewhat, the red color can transfer to the blue and from blue one transfer to the red. As a result, genetically they are changed. Genetically changed means here, so this is blue color and with somewhat red color. As it is another chromosome, red color, somewhat blue color. So this is the combination. So this is the different combination. So this is actually male, apart from the male, female also present. From the parental, father, mother gene, both are segregated in the same chromosome. Here also same. So this is called as recombination. This is called as recombination technology, which is formed generally in meiotic division. But what happened in artificially genetically modified plants? How we get the GM crops? How we get desired crops? Hello?
Sir.
How we get desired crop?
By recombination of DNA only, sir. Hybridization.
How it is happened? First selected the desired gene.
Yes, sir.
Cut the desired gene and enter into the vector. From vector, we transfer it into the host plant.
Host plant.
So this is the process. So in this process, first we have the desired gene, but this desired gene we cut and separate, right?
Yes, sir.
Yes, sir.
So
Like,
this process carried out by restriction endonucleases.
okay.
Restriction endonucleases scan the DNA of the desired gene, recognize the recognition site where they are going to cut and cleave it. And another function can modify the gene, modify the DNA. Right, these restriction enzymes are restriction endonucleases. These are actual enzymes and cut double-stranded and single-stranded DNA. They can cut at specific recognition nucleotide sequences. This is called as a restriction site. Most probably this restriction site in palindromic sequence. What is palindromic sequence?
Polydromic sequence.
Palindromic sequence, both nucleotide sequence in anti-parallel direction. If you read 5 and to 3 end, one stand, another stand, same sequence. Same sequence means here, what do we write? Guanine,
Guanine, adenine,
adenine, thymine, cytosine.
Yes.
So this is 5 and to 3 end direction.
Okay.
And anti-parallel stand 5 and to 3 end direction, right? If you read this direction, G, A, T, T, C. You can read forward and backward, same sequence, right?
Yes.
G from starting 5 and to 3 end. Here 5 and to 3 end, G, A. So this direction same sequence and this direction same sequence. This is called palindromic sequence. Most probably this is the recognition site of the DNA, right? More, and these enzymes, these restriction enzymes are found in bacteria or archaebacteria. And the main function, this provide defense mechanism against invading virus. For example, this is the bacteria. Already bacteria have the restriction enzymes, RE, restriction enzyme. If any chance some other viruses or fungal members, anyone invade this bacteria, invade and enter the host cell. But immediately these restriction enzymes activate and attack the virus and cut the viral DNA. What happens if viral DNA cut into pieces?
Its activity is lost.
If it is DNA is present, what happens in general? This is viral DNA.
The
For suppose Right. For
Sequence get disturbed, sir.
right. For suppose inside the bacterial cell, no are endonucleases. In the absence of endonucleases, when the virus attack the bacteria, immediately the viral DNA produces RNA and like developed protein. Now this protein enzymes attacks the target cell, target part of the bacteria and kills the bacteria. But if restriction endonuclease present, immediately this restriction endonucleases, the main base point of DNA strands cut into pieces. Can it prepare RNA? Can it prepare protein in this situation?
No, sir.
Cannot prepare the RNA and protein. That's why this activity of viral DNA also become deactivated and does not harm to host bacteria. So for this process, mainly inside the most bacteria are having this restriction endonucleases. The first isolated DNA, Hind DNA, first restriction endonucleases is first isolated restriction Hind 2. Hind 2 bacteria here. Hind HIND means Haemophilus influenza. Haemophilus influenza series 2. Haemophilus influenza. So this is the first isolated bacteria. Now let's see the types of restriction endonucleases. Are you clear? First, the basic of restriction endonucleases, function of restriction endonuclease, activity of restriction endonucleases. Now let's see the types of restriction endonucleases. Right? There are actually in our book only given only 3 types are given, but in general 4 types of restriction endonucleases there. They are, please write down.
Okay.
Restriction endonucleases types. First one, type 1. Type 2. Type 3. Type 2s. 2s.
2s.
Right. So there are normally 4 types of restriction endonucleases, right? Clear?
Yes, sir.
These restriction endonucleases important for long question. Types of restriction endonucleases. Type 1, type 2 and type 3. First, let's see about type 1 restriction endonucleases. Type 1 restriction endonucleases. Type 1 restriction endonucleases. Right? Then C5 class జరిగిందా? Hello?
Sir, just now, sir.
Yesterday.
No, sir.
C6,
No.
sorry. Yesterday, C6. Medicinal botany and embryology.
సార్ జరిగింది సార్.
C7
సార్
C7
జరగలేదు
కూడా
సార్.
C7 కాలేదు,
జరగలేదు.
ఓన్లీ C5 రామకృష్ణ
Yes, sir.
సార్ క్లాస్ కాలేదు.
అవును సార్.
ప్రభాకర్ సార్ క్లాస్ అయ్యిందా?
ఎస్ సార్. అంటే ప్రభాకర్ సార్ మధ్యలో ఫీల్డ్ ట్రిప్ లో ఉన్నారు సార్. వచ్చి కొంతసేపు ఉండి వెళ్ళిపోయారు సార్.
Okay,
అంటే
right. సరే. సో రెస్ట్రిక్షన్ ఎండోన్యూక్లియేజెస్ 1. Type 1 restriction endonuclease enzyme is the multi-subunit. It is complex enzyme, multi-subunit enzyme, complex multi-subunit enzyme, and it shows the multitask. As I said earlier, what are the multitask? What are the function of restriction endonucleases? Multifunction.
It protect the host cell.
Before protect, what is the mechanism of the restriction endonucleases?
Producing the enzyme, sir, which will cut the DNA of the virus.
Before cut?
Recombination. It undergo recombinations DNA. Genes.
Recombination, that is the separate thing. First, after cut the DNA, later recombination, that is artificially recombinated by other DNA, other vector. That is different. First, it is before going to cut the DNA, first of all, it is recognize.
Yes,
recognize
sir.
the site, right? Already told you palindromic sequence.
Yes, sir.
After recognize, they can cleave or they can cleave or they can modify. So this restriction endonucleases can recognize the site, can cleave the DNA, can modify the DNA sequence. These three functions done by one restriction enzyme. But this restriction enzyme have the multi-subunit. Multi-subunit means such as multi-subunit are mainly HSDR, and HSDM. So this one also small. HSDM, HSDS. S here small, D small, S capital. So these three subunits combinedly form multi-subunit restriction endonuclease.
Sir, once tell the full form, sir. I'm right.
HSDR.
Yes, sir.
HSDR full form is given HS E. coli bomb, not SOL, POLL, SML, ECO, RV, KPLN, SCACL. Not given the full form of the subunits.
Okay.
This way. Just name of the subunits.
Okay,
these
sir.
are the codes only.
Okay, sir.
Based on the function, the name given as the HSDR, HSDM, HSDS.
Okay.
HSDR is required for restriction.
Okay.
HSDM is necessary for adding
Modify.
methyl group, right?
Methyl.
Modifying.
Yes.
So first, actually, the HSDR restriction means recognize. This is for recognize. HSDM modify. Methyl groups can change the methyl group, and that means modify. HSDS is the important for cut the site. This is this subunit cut the site. So this is the multi-functions of the restriction endonuclease, right? And this type 1 restriction enzyme were the first to be identified and are characteristic of two different strains. K12 and B of E. coli. That means
E.
which
coli
is
bacteria.
E. coli bacteria, which is extracted from E. coli bacteria of two different strains, K12 and B. And another important thing is, actually this bacteria recognition site different and cleavage activity different. Cleavage activity, that means cut the DNA site far away, almost 1,000 base pairs away from the recognition site. 1,000 base pair upstream from recognition site. And this recognition sequence also may be 4 to 6 nucleotides, least 4 and highest nucleotide number 6. Very rare 8 nucleotides present in the recognition site. Let's see once how the recognition site and where the cutting site. This is DNA, right? For example, here this is recognized site and restriction endonuclease enzyme bind here with restriction site. Maybe here this is for this site consists maybe 4 nucleotide or 5 nucleotide or 6 nucleotide. Very rare 8 nucleotide present in this site. Remember, 4, 5, 6, rarely 8 nucleotide present in restriction site only. I am talking about only restriction site, right? Okay. But from upstream, maybe upstream or downstream away from the base pair, 1,000 base pair away, 1,000 base pair away. For suppose this is the area of cut the DNA strand. Cut the DNA strand. From the recognition site to the cutting region, it can scan this all 1,000 nucleotides. The first, this scan the 1,000 nucleotide. After when reach the after 1,000 next nucleotide, where it cut the DNA strand into pieces. So that is the activity. And another important thing is, for this restriction endonuclease 1, several enzyme cofactors, including S-adenylyl methionine, adomate shortcut, and ATP and Mg2 plus ions are required. This restriction endonuclease cannot directly done this activity with help of other recruitments. The other recruitments such as what is the first one? Cofactor and magnesium ion. This and also S-adenosine methionine. Shortcut adomate. adomate and ATP energy also required, and magnesium ion also assisting to this cut the DNA strand, right? So directly restriction endonuclease does not have any activity. For this activity, it required cofactors. It required cofactor, enzyme cofactor, adomate, magnesium, and ATP. With help of all this, scan the DNA, cut the DNA strand. Is it clear first? Hello?
Yes, sir.
Right. So simple from how it is extracted. Multi-subunit, what are the subunits names and functions of subunits and activity. Activity different, recognition, restriction site different, and cutting region also different. And it requires some cofactors and adomate, ATP, magnesium. These are all required for the function of restriction endonuclease 1. Right? Now let's see type 2. This is very important. Type 2 separately may ask short answer. Right?
Yes.
Let's see short answer. Type 2. Type 2 enzyme. This is multi-enzyme complex. Multi-enzyme complex. First one, multi-subunit. One enzyme with the number of subunit, 3 subunit. But enzyme itself multi-enzyme complex. And this multi-enzyme complex, 2 different tasks. Recognition, modification. Recognize and modify, one function. Cleave, cut and cleave, another function. There are no subunits, but multi-enzyme, that means one different task. Recognition, modify, one task, and cleavage, another task. So this is very important in gene cloning. That means these type 2 restriction endonucleases vastly used in gene cloning. And this type 2 restriction endonuclease differ from type 1 enzymes in several ways. The first one, it is not a subunit. It is the complex enzyme. And there are a dimer of only one type of subunit. But what happened in restriction endonuclease? How many subunit? Hello?
3, 4, 4, sir.
Restriction endonuclease subunits 3.
3.
Only 3 sub. But here only one type of subunit. This one subunit, it can recognize the site and it can modify the enzyme sequence. So it can recognize the palindromic sequence, the number of nucleotide length, here also same 4 to 8. Remember, mostly 4. Most probably 4. Recognize site nucleotide number. They recognize and cleave the DNA at the same site.
Okay.
But what happened in type 1?
On different sites.
Different site. Almost 1000 base pair away from the
Away,
recognized
yes,
site, right?
Yes, sir.
But here, where the recognition site present, there only cut the DNA into fragment. And another thing is they do not use ATP or adomate for their activity. What about in type 1?
They use ATP for their.
Yes, they can use ATP and yes, adenosyl methionine, edomate. Apart from that, magnesium ions also required. But here in type 2, no ATP, no ATP and no edomate. No edomate.
Then from
Edomate,
where they'll get the energy, sir? They need energy, no, for.
they use only require magnesium ions.
Okay.
They can use magnesium ion and then they don't need any energy because here the energy required for scanning of DNA. Is there any scanning activity done by R2 restriction enzyme 2? Any DNA scanning takes place by R2 like type 1?
No, sir.
No.
No, here why can't use the ATP energy restriction endonucleases type 2 where the restriction recognized site is present, there only cutting region also present. That's why they don't need ATP energy. But they can use only magnesium ions, right?
Okay, sir.
Magnesium ions as a cofactor. They can use magnesium ion as a cofactor, right? So here the cofactor produce some energy while the cutting activity takes place, only cofactor give energy, right? Next, and these are the most commonly available and used restriction endonucleases enzyme because they are the very easy activity. That means the recognition side same and the cutting cleavage site also same. That's why it is the very simple enzymes to utilize in gene cloning. And these are called as a molecular scissors. What is scissor? Scissoring, cut. So
Cutting.
these are called right cutting. That's why called molecular scissor also. Right. And here the one example, the restriction type 2 example BAMHI enzyme. BAMH1 enzyme. That means BAM bacillus. BAM means bacillus amylopyrosis. 1 means type 1. Right. So this is the example for type 2 restriction endonuclease. Next, type 3 restriction endonuclease. First two clear. First 1 and 2.
Yes, sir.
Right. Now third one. Please be silent, please. Type 3. It is a single enzyme, not a multiple enzyme. This is the single enzyme. Single enzyme recognized to separate non-palindromic sequence. Actually, first two palindromic sequence, but type 3 can recognize, of course, it also recognize palindromic and also non-palindromic sequences. Now they can cut the DNA about 20 to 30 base pairs. 20 to 30 base pairs away from the recognized site. It is almost type 1, look like type 1 endonuclease enzyme. Type 1, what is the difference between type 1 and type 3? It also contains more than one subunit. In structurally, it is also made up of number of subunits like a type 1. And another one is recognition site different and cleavage site different, and require adomate and ATP cofactor for their roles in DNA methylation restrictions. Means it is also like a type 1 enzyme. It can scan the DNA strand. For that purpose, require the ATP energy. For example, here this is the DNA sequence where this recognition site now almost already told you just 4 to 5 nucleotides are there in recognition site. Here the bind with help of restriction endonuclease 3. But the actual activate site here. This is the cutting place. Almost this gap 20 to 30 nucleotides. 20 to 30 nucleotides. Before cleave the DNA, this restriction endonucleases scan the DNA. After scan, reach the site of the cleavage and cleave here. Right? So this scanning purpose, it required some ATP. Same like a type 1, it is also for this activity of type 3 enzyme require ATP energy ATP require, adomate require, cofactors also, whatever it may be, either magnesium, ferrous, any other cofactor also require. ATP, adomate and cofactor. These are all required for the activity of type 3 enzymes. And type 3 also not useful for gene cloning. That means here the difficult to use, but not convenient to us. As well as type 1 also not convenient to gene cloning, but most convenient in gene cloning, type 2 restriction endonuclease. Right? Type 3 clear?
Sir.
Right. Next, type 2S. Type 2S almost look like a type 2. It is made up of two different type of enzyme complexes. It is also same multiple task like a type 2S. It is also multiple task. Almost this function resemble to the type 2 restriction endonuclease. Right? So this is also not important in gene cloning. Only remember gene cloning point of view, the most important enzyme is restriction endonuclease type 2 only. Right? In our book given page number 104 and 105, the example of different type of restriction endonucleases table given. Here just any five enough to write in exam point of view. And the 5 must be the first one, E. coli, Escherichia coli, restriction endonucleases, Escherichia coli endonucleases 2, BAMH1, HIND3. First 5 enough. No need to write this all. In exam point of view, other type of restriction endonuclease, write down and just give 5 sites. And also here the recognition sequence and cut sequence also given another box. First column, enzyme name, second source where they extracted, and third one site of the recognition. Now cut the last column, cleave the cleavage site. Right? Is it clear? I will explain mechanism of cut, blunt and sticky ends. I will explain later. But
Okay,
so far,
sir.
this type 1, type 2,
Okay,
type
sir.
3, mainly type 3. These are all clear?
Yes, sir.
Right.
Yes, sir.
Sir, then no need of writing type 2, yes, sir. Type 2,
No
yes.
need. Type 2, yes, no need.
Okay,
Just
sir.
put the heading in, enough.
Okay, sir.
And just write the one word. The same, it look like a almost similar to the type 2 activity. Enough. similar to type 2 activity. If you want to
క్లోనింగ్ కి
write
యూజ్ అవుతుందా సార్?
what?
క్లోనింగ్ కి యూజ్ అవుతుందా సార్ టైప్ 2s?
టైప్ 2s కూడా క్లాన్ యూజ్ చేయొచ్చు, కానీ దీనంత కన్వీనెంట్ ఉండదు. టైప్ 2 అంత కన్వీనెంట్ ఉండదు.
ఓకే సార్.
రైట్. Nomenclature. Simple, here also just a simple diagram, table given. Abbreviation E for Echerichia. CO, genus, species name, coli. R, type name. One means first identified. Here simple. Always this enzyme names in form of shortcut like this. ECOR1. First one, genus name. Genus. Name of the genus, Echerichia. Second name, species. Species name, coli. Echerichia coli. RY13, strain name. RY13, strain name. One, first identified. Identification in the bacteria. So this is the first identified. Of course, there are from Echerichia coli number of restriction enzymes are extracted, but the first one given name first, second name second, third one third. Like this, number of restriction endonucleases are there, right? Now let's see recognition site and cleavage site mechanism. How it is cut the DNA at the palindromic sequence. Right? This is all clear?
Yes, sir. Okay,
Right.
sir.
Recognition site, cleavage site. Two different ends are there, sticky end and blunt ends. First, take one palindromic sequence. As I said earlier, G A T T C. G A T T C. This is the one sequence. Take the sticky ends cut. First, sticky end. In sticky end, the cut the region is not equal. G. Right? Here same G between GA, here between GA. Guanine adenosine, here also same guanine adenosine. Cut like this. If you cut this manner, guanine separate, and adenosine.
Sir, it is
Adenosine.
cleavage mechanism, sir.
Yes, cleavage mechanism.
Okay, sir.
C, T, T, A, A. And here G also separate. Now this is empty space, right? This side separated, cut and separated. Now it is easily joined by another nucleotide. Another nucleotide means here the same complementary sequence joined this region. Same complement means here adenine, adenine, thymine, thymine. So this desire gene, for example, do you want to take the desire gene? Do you want to insert a desire gene? You cut into the sticky ends. Sticky means what is sticky?
Like gums are attached to one another.
Right, attach one another. Actually, this is only empty. This end only sticky end. Now it can easily bind with another complementary DNA sequence. Another complementary DNA sequence. Because already this region cut, cleaved,
Yes.
separated, and this it is fulfilled with another complementary DNA sequences. Right? That's why it is called sticky end. And another one, blunt end. In blunt end, what happened here? C, 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 G, 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 C, G, G, G. Blunt end means
Which the side of anything is not sharp.
right. No sticky ends.
Yes.
No sticky ends. The cut into two. It is not actually. This is not applicable for rDNA technology. How can recombine? How can recombine in this situation? How can recombine? Recombine now some empty space are need, right?
Yes, sir.
But in case of it is in blunt ends, there is no space for stick or bind by another desired gene. So that's why the most restriction enzyme cut the DNA into sticky ends, right?
Okay.
Yes, sir.
Right. The different restriction enzymes that recognize the same sequence. Here, they actually, this restriction enzyme mechanism, three types. Three types are actually in our books given only two, neosegomers and isosegomers. Apart from that, cardomer, isocardomer also we can mention here. So let's see this mechanism by three ways. Can I rough it?
Yes, sir.
Means we can write in the form of flow chart, no, sir?
Better to write in flowchart enough. Right. What I said, isocysomer, neocysomer, neo, neocysomer, isocysomer, isocodomer. Firstly, neocysomer. Neosystem are different restriction enzymes that recognize the same sequence. For example, this is strains. One enzyme, two enzyme. For example, this is 3 restriction enzyme. But anyway, these are the different type. But this different type of enzyme recognize the same sequence, but recognizing sequence same. For example, sequence may be Tata box. For example, Tata box. Here the recognized site same. That means this enzyme recognize the Tata sequence, this enzyme Tata sequence, this enzyme Tata sequence. And but cleave differently. Recognize same, but cleave differently. Cleave different means your actual Tata box, right? TA. For example, first enzyme cleave this site only. Or second can cleave after TA.
Okay.
And third can cleave after third thymine. So cleavage site different, but recognition same. In neoisomer, recognition sequence same, but it can cleave the different nucleotides. One may be after thymine, one may be thymine adenine common. After thymine adenine can cleave. But another one, for example, thymine adenine thymine. After thymine can cleave the DNA site. Clear?
Sir.
Is it okay? Hello?
Yes, sir.
Right. Next, isocysomer. Same, these are often cleave in different locals. Our different enzyme that recognize and cleave in the same location.
Means like neosysomers only, sir. Same.
No. Isosomers different enzyme. Okay, enzymes different.
Okay.
But recognize same as well as cleave the same. Same like here, what happened? The same different type of enzyme, right? 1, maybe 2, maybe 3. Same enzymes, we can, let me take the same example, TA, right? All are cut the same sequence, maybe after thymine adenine. One can cleave the thymine adenine, 2 can cleave the thymine adenine, 3 also can cleave the thymine adenine, or can cleave the first thymine, or can cleave the second thymine, but are different enzymes, the function same. Have you got it?
Yes, sir.
What happened in neoisomer? In
Yes, sir.
neoisomer,
Means one separately it will get cleavage 1. Here 2. Like.
Can you give the simple sentence answer? Different between neoisomer, isoisomer?
Means in neosysomer singly it will get detached. Means cleavage. In isosomers jointly like couple.
No. Isocysomer, neocysomer, both are different stain. First of all, first point clear.
Yes, sir.
This different enzyme, what I mean neocysomer, recognition site same in both. That means even it is different strain of enzymes, different type of restrictions, but their activity of recognition same in both. That
Okay,
means
sir.
recognition site of the neocysomer, recognition site of the isocysomer same. But what happened? Neocysomer cleavage different. Hello?
Okay. Yes, sir. And I'm getting, sir. I'm getting that.
Cleavage is
Cleavage.
same here.
Yes.
But in isocytomer, iso means same. Isocytomer recognition site same.
Recognition is same, but cleavage is different, sir.
Cleavage
Cleavage
also
also same in isocytomer.
Mhm. It-
Don't get confused.
No, sir.
Neocytomer.
No,
Yeah.
sir. Here, in neocytomer, T after cleavage, A after cleavage. But in isocytomer, here TA cleavage, then TA cleavage. Like,
Whatever it may be, maybe TA or T or last T, anyone, but all three different enzymes function same. Here, first of all, for example, neocytomer. In neocytomer, what happened here? This way. And writing about first type of enzyme. First type of enzyme only can TA. First type of enzyme only cut this region only.
Okay.
If you take another type of enzyme also same, recognition side same, TA does not change, right? But the second type may be cut this region only. Okay. And now I take third type, same recognition site, but cleavage portion this region only. Now clear?
yes, sir. Okay, sir.
Cleavage different.
Cleavage site recognition sequence same.
Same.
4 nucleotide, where you read TA, Tata box, Tata nucleotide. But in neocytomer, the cleavage point different. Cleavage point different.
Okay, sir. Okay, understood, sir.
In isocytomer, recognition site same and cleavage same. If you take the first in isocytomer, first TA, this is the first enzyme activity place. This is a cleavage place. If you take second enzyme, the second enzyme also same, the same region. If you take the third one also same, can cleave this region only. Right? Okay?
Understood, sir.
Right. Now iso cardamar, not given in our book, just had it. Isocardamar, these are isolated different source of their same. These any type neocytomer, isocytomer, isolated from different. They are not similar in recognition cleavage. These three different enzyme. For example, here enzyme A, restriction enzyme A, restriction enzyme B, C. A can recognize Tata sequence.
Recognition is
B can recognize whatever some GOGAT.
same.
This is it. C can recognize what is another one. Just given GAT. Is this recognition same?
No.
No, sir.
No, sir.
Different type
sir.
of recognition site. And cleavage also different.
Different.
Of course, here the different recognized site are different. Right? This is
Understood.
isocardamar. Clear?
Yes, sir.
Right. And again given here restriction endonuclease 2, type 2 enzyme. Just somewhat elaborate, that's it. Type 2 restriction endonuclease, same already discussed earlier, right? So just remember some few words. Restriction endonuclease enzyme, These are called molecular seizures. Already told you. Another name, restriction type 2 enzyme, molecular seizures. Seizures means cutting. And also another name, it is called work horses. Work horses. Work horses of molecular biology, because it is this type 2 enzyme involved in RFLP, DNA sequence, DNA cloning, DNA technique, genome mapping, restriction fragment length polymerism. I will explain later this RFLP,
Restricted.
DNA technique, DNA genomic mapping, DNA sequencing. These are all I will explain later in separate unit. And remember the words here, molecular seizure and work horses of molecular biology. Work horses. This point of view very important in competitive level. Molecular seizures, work horses, right? This type to
work horses, sir. Horses.
S. Horses. HORSES.
Okay.
Type 2 restriction endonucleases recognize short nucleotide sequence. What is the number of nucleotides commonly in recognizing site?
4.
Hello?
Sir?
8.
Number of nucleotides, general number of nucleotide, right? 4.
Five,
Actually
6,
4 to
8.
6.
Yes, sir.
General 4 to 6, very rare 8. And it can give double stranded DNA at a specific site. And it can recognize and cleave the same place.
Same place.
Yeah.
Double stranded DNA at specific site. The length of this recognized sequence varies. That means based on different type of enzymes, the length also different. Maybe 4 nucleotide, 6 nucleotide, 8 base pairs, 8 nucleotide. Here given some example also. The enzyme not NOT, one type of restriction endonuclease to NOT
Restricted
first. And SAU3A1. 2 enough. Another one. Okay. Only 2 given, 2 enough. Not one enzymes. This is recognize 8 nucleotide, and SAU3 recognize 4 nucleotides. 4 base pair nucleotides.
SAU3AI, sir.
And first that one.
Okay.
Roman number 1. The length of the recognized sequence indicate how frequently the enzyme will cut an average in random sequence of DNA. Some enzymes 4, some enzyme 8. Okay. A recognized site will cut every 48 to 60 or 48, 65, 536 base pair.
Space.
That's why these are rare cutters, right? Rare cutters. Rare cutters means, for example, this is the DNA sequence, right? For example, this is the one site. After 200, another site. After 64 or after 300, 500, another site.
Length will be different.
Less, that's it.
Okay, sir.
So this is called rare cutter. But almost this recognized site sequence only 4 to 6. Or maybe rarely 8 also present. But distance between the distance 40, least 48, highest to 500 and 600. This gap, right?
Understood.
Right. And cleavage site ambiguous, no need. Okay. Same cohesive 5 and blunt end already completed. Palindromic sequence that's also completed. Cleavage side may be ambiguous, means should be clear. Cleavage side should be in clear sequence. And next one, here given example XOU. No need these examples. XHO2 type 2S. No need. But last, blunt and sticky ends. You have to write cohesive 5 ends, cohesive 3 ends, blunt end. That means blunt and sticky ends. Cohesive means here sticky ends. Blunt end means
Blunt.
no sharp.
No repeat. No stop.
No place for sticky. There
For
is no place
cleavage.
for another nucleotide sequences.
Sequences.
Right? Is clear? Example avasarama? No need. BPU A1. Okay. In representing nucleotides. Okay. Right.
Finished, sir?
No. Wait. Applications. Application restriction endonucleases. Mainly these are involved in
For desired, to get the desired characters in plants.
right. The given clearly assist insertion of gene into plasmid vector. TA plasmid or DNA. One word enough.
Okay.
To formation rDNA. Right. Another one is restriction enzymes can also be used to distinguish gene alleles by specific recurrence single change DNA. Another one, these are involved in SNPS. I will explain next in it.
Okay, sir.
But here remember, the restriction enzymes can also be used to distinguish gene alleles. That means it can change the gene order. Single base chain. For example,
if the gene order changes, then the characters also changes.
Also change. Right. Character also change. For example, here nucleotide sequence, adenine, guanine, cytosine,
dwarfism,
guanine, cytosine, like this.
Plants.
So this is actual genetic makeup. But due to with help of these enzymes, restriction endonuclease enzymes, it can alter the any one gene sequence. What happens if the gene sequence changed? Automatically, the genetical character also changes. Right? I will explain later in SNPS. That is in single nucleotide polymorphism. Single nucleotide polymorphism. And another one.
Polymorphism means changes only, sir. Meaning?
Yes. Another one, what we said here, just where it is. RE, right. Restriction fragment length polymorphism. I will explain later. These two, restriction fragment length polymorphism, single nucleotide polymorphism. Okay. Right. Another function. These are the involved in gene analysis by Southern blotting. DNA analysis by Southern blottings. It is also involved in Southern blotting technique. Southern blotting మీకు లేదనుకుంటా. Okay, right. ఉంది. సదరన్ బ్లాటింగ్ టెక్నిక్. మీకు సదరన్ బ్లాటింగ్ టెక్నిక్ దాంట్లో ఆర్డిఎన్ టెక్నాలజీ చెప్పేటప్పుడు చెప్పలేదా? చెప్పానండి అసలు ఇది వెస్టర్న్ బ్లాటింగ్ టెక్నిక్, సౌతర్న్
రాలేదు
బ్లాటింగ్
సార్.
టెక్నిక్ రాలేదా? ఓకే, నెక్స్ట్ ఎక్స్ప్లెయిన్ చేస్తుంది సదరన్ బ్లాటింగ్ అండ్ వెస్టర్న్ బ్లాటింగ్ టెక్నిక్. మీకు జీన్ సారీ ట్రాన్జెనిక్ ప్లాంట్స్ దీంట్లోనే కదా, జెనెటికల్లీ మోడిఫైడ్ ప్లాంట్స్
సార్
టెక్నిక్,
ఆ టెక్నిక్
జీన్
లో కూడా రాలేదు.
టెక్నిక్, జీన్ టెక్నిక్స్.
ఓకే, ఎక్కడ వచ్చింది?
ఈఎంఎస్
ఎక్కడ వస్తుంది ఇది? జస్ట్ వెయిట్. మిథైలేజ్ క్లోనింగ్ వెక్టార్.
సార్ క్లోనింగ్ లోనే జీన్స్ టెక్నిక్ ఉంటుంది కదా సార్.
సిడి అని మీకు ఎక్కడ ఉందండి జీనోమిక్ లైబ్రరీ. పాలిమరేజ్ చైన్ రియాక్షన్, రైట్. మాలిక్యులర్ మార్కర్స్ లో రైట్ అండి, మాలిక్యులర్ మార్కర్స్ లో వస్తాయండి ఇవన్నీ. ఐ విల్ ఎక్స్ప్లెయిన్ దేర్. ఓకే,
ఓకే సార్.
but remember the word southern blotting, western blotting technique, RFLP technique, and another one is SNPS technique.
SNPS.
Right. So these are the main functions of the restriction endonucleases enzymes. No need to write these are all in detail. Just 5 points enough for application, because the application never asks for the short answer and long answer. This is only along with the restriction types. They can ask write about restriction endonucleases with applications. That's enough. In this situation, just you have to highlight the type of restriction enzymes, right?
Okay sir.
Ligases, unit number 8, modifying enzymes in molecular cloning. Let's see first ligase enzyme. Complete opposite to restriction endonuclease. Restriction endonuclease cut the sequence. Ligase enzyme join the sequence. That's it.
పేస్టింగ్ సార్.
What? Yes, that's it. Paste. Here same, but some restriction enzyme need ATP, but some other not use ATP energy. But for in ligase mechanism, they should need ATP enzyme, ATP energy required for join to different fragments. Actually, different fragments means fragment. This is also fragment. Phosphodiester bond 5 and 3 and 5 and phosphate and 3 hydroxyl group. For example,
Hydroxyl means OH group.
OH group.
Okay sir.
OH group, third carbon, triphosphate commonly. So the bond formation hydroxy group and phosphate group of 5th carbon, third 5th carbon. Giant. Actually, when you cut the DNA, these phosphate groups are removed. That means this region is separated. With help of ligase enzyme, these two again rejoin.
Yes sir, it converts like ATP to ADP and ADP to ATP.
Just wait. Right. That means after that, this ATP convert into
ADP.
Just wait. I will explain. Just wait. Where it is? Are you able to see the screen? Just load. Hello?
Yes, sir.
Right.
1 second, sir. We'll take screenshot.
Just wait.
With 5 end, 3 end. Hydrogen bond.
So here 5 phosphate. Giant, this is 3 hydroxy group here. Hydroxy group. 3 hydroxy group, 5 phosphate. Now the phosphodiester bond formed between 3 hydroxy group and phosphate. Right?
Yes, sir.
Clear? Okay, rest of the class I will explain later.
Okay, sir.
Ligases and phosphatases, nucleases, methylase, these are all. Right. Any doubts so far?
Till now, no, sir. Okay, sir.
What about other subject? C5, C6, C7?
Going through the textbook, sir, whatever the sir teaching.
అర్థం అవుతున్నాయా క్లాసెస్? ఏమైనా
అర్థం
ప్రాబ్లమ్స్
అవుతుంది
ఉన్నాయా?
సార్. అర్థం అవుతున్నాయి సార్.
ఓకే.
సునీల్ సార్ వాయిస్ కొంచెం వినిపించట్లేదు సార్, కొంచెం ఆడబుల్ గా ఉండట్లేదు, కొంచెం బ్రేక్. సునీల్ సార్
రైట్. ఓకే. ఇంకా ప్రభాకర్ సార్, రామకృష్ణ సార్ అన్నీ ఓకేగా క్లాసెస్?
ఓకే సార్, నో ప్రాబ్లం.
అంటే క్లాస్ ఓకే అంటే క్లాస్ ఎండ్ ఆఫ్ ద క్లాస్ వచ్చేసరికి మీ అవుట్కమ్ కి ఆ క్వశ్చన్ నేర్చుకొని రాసేటట్టు అవుతున్నారా? కరెక్ట్ గా చదివేది అంటే బుక్ క్లోజ్ చేసిన తర్వాత, అంటే క్లాస్ అయిపోయిన తర్వాత బుక్ ఓపెన్ చేసి చదివితే మళ్ళీ ఓన్గా నోట్స్ ప్రిపేర్ చేసేటట్టు ఉండాలి.
మేము టెక్స్ట్ బుక్ పెట్టుకుంటున్నాం సార్ రన్నింగ్ అయ్యేటప్పుడు క్లాస్.
రైట్. ఎప్పుడైనా మీరు క్లాసెస్ కంటిన్యూ అవుతున్నా ఒకవేళ అడగాలనుకుంటే అప్పుడు స్టాప్ చేసి అడగండి, లేదా ఇబ్బంది ఎందుకంటే అయిపోయిన తర్వాత టైం ఇస్తారు. అందరికి చెప్పాను నేను ఆల్రెడీ, ప్రతిసారికి ఇన్ఫార్మ్ చేశాను. లాస్ట్ ఒక 5 minutes వదిలేయండి డౌట్స్ క్లారిఫై కోసం అని చెప్పాను సార్ వాళ్ళకి. ఎందుకంటే ఇది లైవ్ కాబట్టి మనకు డిస్టర్బ్ అవుతుంటుంది మధ్యలో అవుతే, అందుకనే కొంచెం లాస్ట్ టైంలో మనం డిస్కషన్ పెట్టుకున్నాం అనుకో క్లియర్గా ఉంటుంది. మీరు అప్పుడు నోట్స్ రాస్తుండండి. క్లాసు రన్ అయ్యేటప్పుడు మీకున్న పాయింట్స్ అన్న అక్కడ నోట్ చేసుకోండి. అయిపోయిన తర్వాత 5 minutes ప్రతి ఒక్కరికి చెప్పాను, 5 minutes టైం ఉంటుంది, ఆ 5 minutesలో 10 minutes ఇచ్చేయండి, ఆ టైంలో క్లారిఫై చేయమని. ఓకే కదండి. ఇప్పుడు ఈ సెకండ్ ఇయర్ గ్రూప్ అడ్మిన్ ఎవరున్నారు? వాళ్ళ నెంబర్ కలుస్తలేదు, నా నెంబర్ ఎట్లా ఎగ్జిట్ అయిపోయింది. ఇంకెవరు ఉన్నారండి అడ్మిన్గా?
తెలియదు సార్, నేను లేను. అంటే గ్రూప్ లో ఉన్నా కానీ అడ్మిన్ కాను సార్ నేను.
ఓకే,
లత అనుకుంటా సార్, లత వాళ్ళు.
లత ఆర్ స్వర్ణలత.
స్వర్ణలత ఉన్నారా గ్రూప్ అడ్మిన్ ఉన్నదిగా? ఆ నెంబర్ ఉంటే ఈ నెంబర్ నా నెంబర్ యాడ్ చేయమని చెప్పు వాళ్ళకి. ఎట్లా మిస్ అయిపోయింది నిన్న పిల్లలు ఆడుకున్నప్పుడు.
ఓకే సార్.
ఓకే సార్. ఓకే
ఓకే ఓకే అండి
సార్. ఓకే సార్. గుడ్ నైట్ సార్.
ఓకే, గుడ్ నైట్ సార్.
రైట్ అండి, బాయ్ బాయ్, గుడ్ నైట్
సార్ గుడ్ నైట్.